Welcome back to previous year IIT JAM Biotech Solution series. In this video, I will going to solve numerical problems that appeared in 2017. So let's see how to solve those problems stepwise. In this problem, three amino acids X, Y and Z are mixed and subject to electrophoresis. The pH of electrophoresis buffer is 5. The isoelectric point of each amino acids are provided and you have to predict what will happen after electrophoresis. When the pH of the solution is acidic, the amino acid will adopt this structure. So at acidic pH, amino acids are positive charged. The amino acid will remain in this form until its isoelectric point reached. For example, if the pI of an amino acid is 6, then at pH 6 it will adopt this structure. It becomes neutral, but below pH 6 it is positively charged. When solution is basic, it will adopt this structure. That means negatively charged. Now come back to the problem. Here we will look at each amino acid one by one. First, we will watch amino acid X. Its pI is 9.87, means it will become neutral when pH is 9.87. But electrophoresis is undergone at pH 5. So X will be charged at pH 5. Now Y, its pI is 3.22. So solution is basic compared to its pI. So amino acid Y will be negatively charged. Only amino acid Z will be neutral because its pI is 5.43, that is close to pH 5. So Z will not move towards any electrode, X will move towards negative electrode and Y will move towards positive electrode. So option A is correct. In the second problem, standard free energy change of glucose to lactose conversion is provided and ADP to ATP conversion is also provided. And you have to calculate the energy efficiency of homolactic fermentation. We know that during homolactic fermentation, 2 molecule ATP are produced. So 2 into 30.5, that is 61 kilojoule per mole energy is captured in form of ATP and rest is released as heat. So in terms of ATP, lactic acid fermentation is 0.31 or 31% efficient. So option A and C both are correct. Now in this problem, interaction between antibody and antigen will result antigen-antibody complex. The rate constant for forward and backward reaction are provided and you have to calculate the dissociation constant. We know that dissociation constant can be calculated from this equation and here it is 4. You can also remember this equation for association constant calculation. Here in this problem, a bacterial population is increased from 1000 to 1 billion in 5 hours. You have to calculate the doubling time in minute. Here initial number of bacterial cells is 1000 and final bacterial number is 1 billion and time t is 5 hours or 300 minutes. Now you have to calculate the g using this equation and here g is 15.05 minutes. Next, the value of Vmax and Km and substrate concentration are provided and you have to calculate the initial velocity in nanomolar per second of this enzyme substrate reaction. To solve this, we will use Michaelis-Menten rate equation. Here substrate concentration is 0.25 millimolar, that means 0.25 into 10 to the power 6 nanomolar. Maximum velocity Vmax is 5 nanomolar per second. And Michaelis-Menten constant Km is 1 millimolar or 1 into 10 to the power 6 nanomolar. So V0 is 1 nanomolar per second. In the next problem, a linear DNA contains 5 restriction sites for EQR1 and 3 for BAMH1. But you have to calculate the number of fragments will be generated after EQR1 digestion only. We know that in a linear DNA, if the number of restriction enzyme sites are n, then the number of fragments will be n plus 1 and here the DNA is linear, so the total number of fragments will be 5 plus 1, that is 6 after EQR1 digestion. In this problem, the standard oxidation potential of NADH and water are provided and you have to calculate the standard free energy change in kilojoule. First, we will calculate the standard oxidation potential for oxidation of NADH and H2O and it is 1.13 volt. So the standard free energy change can be calculated using this equation. Here N is 2 cause one molecule oxygen required to oxidize one molecule NADH. F is 96.5 kilojoule per mole. So the standard free energy change is minus 218.09 kilojoule. In the next problem, in presence and absence of inhibitor, the value of Km and Vmax are recorded and you have to calculate the value of inhibition constant in millimolar. First, we will check what type of inhibition it is. Here Vmax remains unaffected in presence of inhibitor, so it is a competitive inhibition. In competitive inhibition, the value of Km which is observed in presence of inhibitor is called apparent Km and denoted by alpha Km, and here it is denoted by K prime M, and it is 6 millimolar. So change in Km in presence of inhibitor is 1.5 fold. Again we will use this relationship to calculate the value of Ki, and here Ki is 3 millimolar. In the next problem, a relationship between log of molecular weight of a protein and RF factor is established using this relationship. You have to calculate the number of subunits in a protein in which 180 amino acids are there. The RF value is also provided. We know that the average molecular weight of amino acid is 110 Dalton. So here the molecular weight of the protein with 180 amino acid will be 19.8 kilodalton. Now we will calculate the log of molecular weight of the protein if the RF value is 0.5 and here it is 2. So the measured molecular weight of protein is antilog of 2 or 100 kilodalton. Now we will calculate the number of identical subunits and here it is 5. In the last question, a bacterial ribosome takes 20 seconds to synthesize a protein with 300 amino acids. The average lifetime of a mRNA is 2 minutes and you have to calculate the number of ribosomes that can translate 1350 nucleotides long mRNA. So in 20 seconds, ribosome can synthesize 300 amino acids or it can translate 900 nucleotides. So in 2 minutes or 120 seconds, it can translate 5400 nucleotides. But the size of the mRNA is only 1350 nucleotides. So the number of times ribosome will translate this mRNA is 4. I hope you find this video helpful for your JAM exam preparation. So subscribe my channel and don't forget to press the like button and make your comments. And thanks for watching.